And I have a feeling if any real biochemist looks at this, or biologist, they're going to flip out at me and saying something wrong, which is fine. They can tell me later. So cells in a resting state. When we went through and looked at this, we said our correct answers were high ATP to ADP ratio and high ratios of oxaloacetate to citrate. Okay? So high ATP and ADP. So in a resting state, we don't want Krebs cycle running. The way we shut down Krebs cycle, high ATP. So it makes sense to have more ATP in there. If Krebs cycle is shut down, what happens to the alpha-ketoglutarate enzyme? It's going to turn off. So that should be inactive, okay, not active. Okay, so that doesn't or shouldn't be true. High ratios of oxaloacetate to citrate. Okay. Well, our conversion from oxaloacetate to citrate was our on-ramp into the Krebs cycle. Acetyl-CoA dumped in and converted oxaloacetate to citrate. If we're saying we don't start the Krebs cycle, we should have a lot of oxaloacetate and virtually no citrate.
But isn't that used as an intermediate in other stuff, or used in other ways? You would think that if it was in a resting state, you would be taking this oxaloacetate, using it in other.
And so this is why it's nice to bike. Because in that hour bike ride home or the hour bike ride in, I'm going through like, I don't like the answer to that. What's going on with it? What are different approaches? How can you look at it and see what's going on with that? And I completely agree with that statement. Okay, and so this question came up because I had to have found it somewhere. So someone went through and said, this is the way you should be thinking about it and looking at it. If we go through and look at a complex biological system, is the Krebs cycle the only thing happening? No, it is not there just to produce energy. It is there to produce intermediate molecules that can then go out. We don't want to look at catabolism. We want to look at anabolism. Come on. Those intermediate molecules can, where's the nice one? There it is. They can then go out and make a variety of things. So when we're looking at a resting state, what happens to all of those molecules that have now been made, or all of those intermediates? Okay, well, they're just going to be sitting there doing absolutely nothing. Okay, well, does that make sense? Look at all those other pathways. If we're in a resting state, now's the time that I can go through and build extra stuff. I can take that excess energy in my ATP. I can take that excess individual substrates within that mechanism and feed those out to do something else useful. Okay? And that's the argument that Jimmy's trying to make here, I believe.
Yeah.
Okay? We're saying that, okay, we're in a resting state. I don't want Krebs cycle to run. Okay? So if we ignore kind of the peripheral stuff, okay, oxaloacetate concentration is going to build up slightly in comparison to the citrate, because we'll finish out the citrate and get rid of it, and then we'd have a bunch of oxaloacetate left over. Okay? And that's the argument that the question is making. But then what you're then suggesting, Jimmy, is that, well, it's not just generating energy. Those molecules can be, or those intermediates can be used to make something else. Okay? So yes, I may complete the cycle of citrate into those individual molecule or the other intermediates, but then I now have a bunch of stuff chilling left over. I can start converting oxaloacetate into aspartate. I can convert aspartate out into nucleotides.
Well,
right? So
that
what's going to happen to the concentration of oxaloacetate?
it's going to continue to go up because you said it wasn't controlled by ATP or NADH, but it continues to go up.
But if we look at our citric acid cycle, our oxaloacetate concentration should not continue to go up at resting state.
But it doesn't get shut down with NADH or ATP, so even in resting, it would continue the citric, or the acid cycle continue up all the way to oxaloacetate on that side of the citric. You know, like with the
But
other,
that assumes you're still producing pieces. That assumes acetyl-CoA is still getting dumped in. That assumes you still have citrate.
But I thought you said on the right side it was controlled by NADH and ATP, but on the left side of it, it's not. And even though you say that like malate can go out and it can produce amino acids, malate also can come in and go back up and form acetyl acid.
And this is where it turns into a big nasty
So
mess of weave of what's going on with it. Okay, when we look at our reactions, okay, everything that's happening in here is in equilibrium. And we look at biology in general. We get changes when we shift out of equilibrium, but the standard state for all life is equilibrium, which means what happens to our concentrations? They should all stay the same. Okay, nothing should really vary outside of that. And finding that exact balance on where that equilibrium lies is a different question. And that, unfortunately, we don't have enough context to evaluate. So when we look at our resting state, we have achieved an equilibrium. Okay, what happens the instant we become active?
We're out of equilibrium.
We're out of equilibrium. Now things have to start to kick in. Okay, and as soon as we become active, what happens?
Then it turns it into citrate. It's going to take the...
We need energy. If we need energy, what happened to the concentration of ATP?
It goes down.
It went down. So now our ratio changes between ATP and ADP. We're losing ATP. We need to regain it. How do we regain it? We kick on Krebs cycle.
So would you say then...
So then all sorts of things start shifting and going out of whack. Well, it's going to change everything. Okay? At the instant we now have no ATP, what happens to Krebs cycle? Krebs cycle becomes active, which means alpha-ketoglutarate becomes active, which means the whole cycle starts up and starts running, which then means our oxaloacetate, as soon as it's formed, consumed and back into citrate. So our oxaloacetate to citrate ratio should start to adjust, where our oxaloacetate starts to decrease and our citrate should start to increase. Okay? What was the initial state?
We don't know.
We don't know. But we can say the relative concentrations of those would change the instant we go into an active state. Okay? Once we are in that active state, we have now achieved a new equilibrium, where we are constantly trying to make this energy source. Now what happens as soon as we go out of being active? We'll have a lot of energy left over, because now the system's like, well, I don't need that energy anymore. I'm not going to produce it, or I'm not going to consume it. So ATP is now at an elevated state. Okay? ATP then shuts down the cycle, and our ratio of each of those concentrations will now shift again. So instead of saying which one is larger or smaller, as the question was kind of, I don't know if I'm going the right direction. I think I am. Instead of saying which one was larger or smaller, we should be looking at how it's changing. Okay? When we go from an active state to a resting state, what should happen to our ATP relative to our ADP? Okay. Got to be a little bit careful. The active state, we're consuming energy. What happens when we're in the resting state?
Oh, ATP goes up.
Our ATP concentration is going to appear to be much higher. Okay? Because it's now no longer getting consumed. Okay? What happens to our alpha-ketoglutarate? Well, with ATP being high, that can shut down the alpha-ketoglutarate, so it should be inactive. Okay? So B doesn't work. What about C? What happens to our oxaloacetate to citrate? Well, if Krebs cycle shuts down, okay, oxaloacetate is no longer being consumed, but citrate is. So the citrate concentration goes down. What happens to the oxaloacetate? It's necessarily going to go up if it completed that cycle. So looking at how they change makes sense. Looking at the absolute ratio, I don't think makes sense.
You could say higher ratio, maybe than
That
high
might
ratio.
that might be a better way to look at it, is putting in that qualifier. Okay. Either way, I think it's a dodgy question. Okay. So I won't ask that. What I might...
That's bad, because we know the answer now.
Well, but see, here's the argument that I would go with this, is that if we're in that kind of balance on how we're shifting immediately out of those stress states to relaxed states and back and forth, okay, the rest of the cycle is still going on. Other stuff is still happening. Okay? That stuff is going to be adjusting those concentrations. Okay? So we can't just say, well, just because Krebs is turned off, that oxaloacetate concentration goes up. Because as it goes up, we now have a ton of oxaloacetate, and what does the system do? Find somewhere else to use it, so the concentration drops back down. So you're really kind of answering this question on the threshold of the instant it's changed. Instant it's changed, what now happens? Okay. We aren't looking at the, oh, well, now I have a whole bunch of it. I need to do something with it. Okay? Because that's kind of your later state. If we think about it kind of like our enzyme kinetics, this question, if we look at our enzyme kinetics, right, we reach and achieve a steady state out at one end, and then at one end we have first order dynamics. This question is really asking what's happening in that section. The instant I change, what goes on? Well, a lot of stuff is going on. Okay, so we're seeing some big changes. We can kind of make some sweeping gestures. But what happens up here? Okay, and that, I think, isn't clear in the way the question's asking, or the way the question's phrased. Okay, to really get at it, you have to look at that instant I'm switching from stressed to relaxed, or relaxed to stressed. The instant that happens, that's where I need to be answering my question. Because if I give it any longer to kind of equilibrate and say, okay, I'm now maintaining this level, we're now achieved up in that upper realm, all sorts of other stuff is going to happen along with it. Okay? That's where anabolism can come in. That's where catabolism can come in. And that's really what Jimmy was getting at. Our oxaloacetate concentration goes up. Well, crap, if it's up, I need to do something with it. Same idea with ATP. Do we really want a ton of a high energy molecule just chilling inside our body? No. We want to get that used and put into something useful. So even with ATP, if I have a lot of ATP left over, okay, in our resting state, just sitting here, it's not like the ATP is now just chilling, waiting for you to get up and start running out of the classroom. Okay? It's being used to do something, which means what happens to the concentration of ATP? It goes down. Okay? So as soon as we have it, something's going to happen to it. That's that big, massive cycle of everything that we know about biochem. Okay? When we're being asked a question like this, we're asking at the instant we're changing, going from a stressed to a relaxed state, okay, which becomes a tricky question. I would make the argument that that kind of question is something a standardized pre-health test would ask. Okay? I would not be surprised if you saw that on the MCAT. Mike, you were working on the MCAT stuff. Do they ask a question like that?
There's a lot of questions.
Yeah, no, it's okay.
I'm sure they will.
Okay, so that kind of level, and it's confusing because as soon as you start to process it out and say, well, what happens when we have too much of this? Well, the other reaction is going to carry, and that's going to start to cause things to change and level out. So when you're being asked a question like this, they're asking for the instant it changes. Okay? And the reason this all made me happy when I looked at the practice exam this morning is that when we talked about this, we said the answer was A and C. And I was like, why are two answers right? Like, this seems weird. I didn't really like that. Okay? But if we go through and take a look at the practice exam, oh, stupid. We take a look at the practice exam, question 26. Cells in a resting state would be expected to manifest. Let's go through and work through this. So number one, A. We're at a resting state, what happens? The instant we transition from active to resting. Tons of ATP. We stopped using that energy source that's getting produced, so we're going to expect a very high or a higher amount of ATP relative to ADP. Okay, so that makes sense. Okay, what about the next answer? Okay, alpha ketoglutarate dehydrogenase produces NADH. NADH produces ATP. Do I want more ATP? No. Okay, so I want an inactive form, so that doesn't make sense. Okay, high ratios of oxaloacetate to citrate.
You do those.
Oh crap! Oh wait a second. I thought I'd switched it. Did I start editing questions? That is the same question.
We should have done the practice exam before we went.
Because I can tell you what the answer is on the key.
It's just A.
It's just A. which then becomes questionable when we look at our oxaloacetate citrate for that one. I thought it said low when I looked at it this morning. So give me a quick second here.
To the citric acid.
Okay. If we have a bunch of energy already, NADH, remember NADH will get us ATP in the electron transport chain. Okay. So if we already have a bunch of energy, do I want to initiate Krebs cycle? No. So if I have my molecule of NADH, that can shut it down saying no more energy. That's a solid answer. I don't want any more energy. Don't give me any more. Okay, so NADH can shut it down because we're saying no more energy. What's the other big reason? And the question kind of gives you the background information necessary to do that.
Well, so if it's allosterically controlled, then what we're saying is when NADH binds to this particular enzyme, that it's going to enhance oxaloacetate being changed to malate?
To citrate. And allosteric just means that it changes shape. It does not mean it changes shape to make it work better. Okay? That would be a positive allosteric interaction. It could just as easily be changing shape to screw it up. Okay? Which is actually what's happening here. Okay? And that's not actually being directly stated. Okay? What is that allosteric control? In this case, that allosteric control turns it off. Okay? And we said one answer is NADH is energy. Okay? If I have a bunch of NADH floating around, I don't want to make more. The whole point of our Krebs or citric acid cycle is to make more energy. If I have a ton of energy, don't start it. It's as simple as that. Okay? And NADH is our form of energy, or can be converted to it. Okay? So I don't want to produce more. So NADH can feed back in, turn it off. So it's an energy thing.
What's the other thing?
Okay? What else does the question give you?
Citrate isn't involved in a lot of other anabolic processes, so you wouldn't want to form it unless you want energy.
Krebs cycle isn't just energy. The molecules generated or made within the Krebs cycle can be used elsewhere. Can be. Citrate, according to the question, isn't used. Okay? Oxaloacetate is. If I go into start this reaction, I've converted a useful molecule into a piece of junk. Okay? And that piece of junk can only get me energy. Well, if I don't want energy, that's a pretty horrible thing to go through and do. So I can take the oxaloacetate instead and shuttle that somewhere else in my cell, and I can make stuff. Okay? So it's looking at kind of the two functions on where we can go out of this. One part is looking at what is the goal of citric acid cycle. Citric acid cycle is to produce energy. The other part of it generates these molecules that can be used in other processes, like anabolism. Kind of makes sense. When I add a thing like there are at least two, it's because I can only think of two, and I want to make sure that those two answers show up. Okay, so if you gave me other answers that are still valid, does anybody know any other answers? I can't think of any. Okay, but if you gave me other ones and they were valid and I agree with you, I'd give you points. Okay, but I'm putting that statement there because I want you to think a little bit harder than just the energy concern. Make sense?
So are you going to go? Okay, just keep going.
That's all I was planning on doing. Unless you guys have specific questions you want to get to. We do have 45 minutes left, right? Yeah. You guys can push me to go to certain questions if there's other questions that you're like fine with.
No, keep going.
Okay. We've got NAD+, and we're looking at the...
You flopped up, didn't you? Isn't NADH the first one?
I did.
Because you're on, right?
Yeah. Yeah, that's weird. I don't know why I did that. Okay. Yeah, whatever.
So would we just show that it did resonance then?
Use
So,
positive on the nitrogen.
what we're saying is we have NADH here,
Yeah.
okay? Notice no positive charge, which means we're going to NAD plus, okay? So, yeah, it's definitely phrased weird. I can get out of it because I phrased it weird enough that it works. But I should have phrased it a little bit cleaner. So we want to get to NAD plus.
So nitrogen has positive and we have to change the double bond.
So the nitrogen needs to become positive. How would it become positive?
Because you're going to lose a hydrogen.
Okay, so
For
a hydrogen is going to disappear from where?
fourth carbon.
I don't know how you're where the first atom is.
Nitrogen.
Okay, I'll accept that. So we're saying this is our fourth carbon. I didn't want to give anything away on that. Okay. On our fourth carbon, we're going to lose a hydrogen. A hydrogen is what? H plus, H minus. It has to be H minus. We're going from NADH to NAD plus. So we will lose a hydrogen. So identifying what hydrogen and then identifying how it leaves is going to be important. It is leaving with the electrons. So if we go back to mechanisms, what we're saying is that hydrogen takes the electrons. And this is where everybody in organic chemistry should flip out, because there's no way hydrogen can take electrons with it. Okay? It is reacting in a way where that hydrogen with those electrons is being dumped onto another molecule that can accept those electrons. Okay? So it is not leaving on its own, it is going with something, which is why it's allowed to come off as H minus. Okay? So the way I would approach this is focusing on that. But now what happens to carbon number 4? It would be positively charged because it lost electrons. So we can show in purple kind of our intermediate states. Okay? But you're right. You said the nitrogen needed to be positive. Okay? Well, the carbon doesn't want to be positive because my organic students now might remember. Okay? I've made a bigger deal about it now this semester than I did for your class. What's the issue with that carbon? So if we just drew everything out the same, okay, don't touch anything else. Okay. All we did was get rid of H minus. That is perfectly valid. Why not? Okay. That works. We don't show that as our NAD plus form because there's something wrong with that structure. That thing that is wrong with it
Said it was with resonance.
So resonance is an important issue here. That carbon or that carbocation is missing an octet. Okay. For those of you that vaguely remember, where does octets come into play? Yeah. Where does octet as a word come from? 8. Okay, what do we tie it to in organic chemistry or in chemistry?
The electrons in the center.
In Lewis structures.
Oh.
With it missing an octet, that structure, while a potential structure for this, is a bad structure because we don't have the octet. So what do we end up doing? We go back and say, okay, well, I need to fix it. It's missing an octet, so I'm going to move electrons towards it. Okay. The result is that I would now have the double bond here. And I still have that double bond. But now what's the problem?
And the nitrogen, you gotta get.
That carbon is now missing an octet, so it is equally a crappy structure. So we need to fix that. How do we fix that? The nitrogen donates its electrons over, and what we end up with would be this final structure. Okay. Couple ways that this question could come out would be kind of along these same lines. Do you know the structures across the board? What does NAD plus look like? What does NADH look like? The other thing that I could easily ask, give me the mechanism. Okay? Show me curved arrows that explain how this is working.
So we can do curved arrows.
You can do curved arrows. Yeah. Okay. And if you've memorized your initial and final states, then it's just a question of add curved arrows until you get there. Right? And as long as you know how to move electrons, which you should from organic, okay, and the basic review that we've had, you should be able to go through and process that. Does that make sense? Okay, or let's say it this way. You are responsible for being able to do that. Okay. What's an easy way that I could be like, I don't want to write a new question, but I still like this question. How do I fix it? Go from the oxidized to the reduced form. Okay? So make sure that you can flip it on its head and do the reverse reaction. Does that make sense? Questions there?
So, when I did this, I just went the other way. I took the electrons from the bond on the right and moved them over, and then did that. So then
So it doesn't matter whether you use the left side double bond or the right side double bond, you'll end up with the same structure. You'll notice the double bonds are located in offset by one, but you have an aromatic structure. There's resonance. The double bonds are on equally shared between every single atom within that. You would sometimes see this drawn as just a circle in between or in the middle, saying the double bonds are all the way around. Okay. As long as you got me to that structure, I would accept it. In this case, if I added curved arrows, showing me the circle is a bad idea. You should actually show where the double bonds show up or shift from. Make sense? Major changes: glucose to D-glyceraldehyde 3-phosphate. That's a fun one, isn't it?
So can we do like glucose plus ATP with an arrow, or hexokinase goes to glucose 6-phosphate plus ADP and do that, or do we have to draw?
You can break it all the way down. So in this case, I'm not necessarily asking for structures. I'm asking for changes. Every time we've looked at a reaction, we've identified the changes without doing structures. You don't need structures to go through and evaluate the changes. So
you can just say that the phosphate was added and then it was cut out.
okay. We could say phosphate added. Absolutely. What else could we say?
Carbon 3-4 broken. Like, can you say that? Is that really the
Carbon 3, 4 bond broken. Anything else?
that's what you're saying, that an isomerization takes place. I guess you'd say that, but it's not being very specific.
I'd be fine with just saying an isomerization. Okay. Technically, there's 2 isomerizations, because when you split that in half, you end up with dihydroxyacetone and glyceraldehyde. So you do an isomerization from the acetone to the glyceraldehyde. Okay. So there's actually 2.
2 phosphates being added then.
Okay. Yeah, sorry. And 2 phosphates. Okay. We got the isomerization, and technically in our isomerization, one is a mutation, because you use a mutase versus an isomerase, but it's still doing the same thing. In fact, I think it's the acetone to the glyceraldehyde that's the mutase. Okay. That's still an isomerization. Okay. So in that list, you've generated 5 things that happen. How many steps are there to go from glycolysis or from glucose to glyceraldehyde? Is it 5? I was going to say 6 or 7. Do you actually have it nailed down?
It happens on your 4th step, right? And then that second isomerization step
Where's my arrow?
Well, 'cause you gotta go to glucose to
Looks like
glucose
five.
6-phosphate to fructose, fructose to.
Phosphorylation, isomerization, phosphorylation, cleavage.
Oh, you guys are really right. You suck. 1, 2, 3, 4. Yeah. Okay, well, show offs.
Is that 5?
That's 5.
So it just depends on how many you shove into a step though, because you could break them up in
Where did, oh, 'cause I want this. And and it freaking says 5
Well, 'cause
changes.
There's 2 phosphorylations.
Yeah, so there were 2 phosphorylations, which we listed. There were 2 official isomerization, and then cleavage of the 3,4 bond, which was 5 possible changes. So good, you guys actually listed them all.
So that's really what you want, is
Yeah.
just that phosphorylation, isomerization?
All I want.
Not,
I don't need it. If you want to give me individual steps, you are fine to do that. And you could give it as names even. Okay, if you've got them memorized down as names, you go from glucose to glucose 6-phosphate, cool. Run away with it.
okay, okay.
Okay. Well then glucose 6-phosphate to fructose 6-phosphate
Yeah.
to
Fructose 1,6-phosphate.
yeah. So you're more than welcome to run that all the way through. Okay. But what I'm looking for are the big changes, because that to me is important. Looking at the name of the structure, you can get that in biology. Make sense? Identify the following parts of FAD molecule, the reductive part, the Flavian Mononucleotide and AMP. Okay. Just kind of an annoying question, but fair game.
I don't remember those names. What the names of those?
Those all, and I can even pull up the slide that shows it. Unfortunately,
Yeah, but we never really.
yeah. Well, we never went over it. We talked about each of those pieces.
Yeah, I like to remember. what
Nice
they
try. Oh, come on, stupid. So it's acknowledging each of the pieces within it. Okay. The other thing we can do is just work from what we've got. Okay. So, which of those parts do you have any ideas on? What is AMP? Our AMP is our bottom. Okay, that would come from having looked at ATP and recognizing the structure of ATP. You know what the A is. We've got our monophosphate. There should be a P in there. Sorry. Okay. And then we have the reductive part and the Flavian Mononucleotide. The reductive part, similar to NADH. Shoot, did I circle the right ones? I think I went did I do the right ones?
Yes, because that's it.
Yeah, because we don't go to the other nitrogen. Yeah, we need to put on 2 hydrogens. So there's our reductive part. Okay, the one part that, and actually it was kind of nice there. The Flavian mononucleotide could also be written as FMN. If we think back to what we saw in the electron transport chain, there was an FMN. It's this. So your flavin is that part of your nucleotide. That's technically the nucleotide. If you attach that to a sugar and a phosphate, you actually have a nucleotide. So your nucleobase nomenclature comes from DNA kind of stuff. We haven't talked about that, but that's what the nucleotide is referencing. Okay, so this is our FMN. Okay. Easy way to switch that. Do NAD. Do FAD, or that is FAD. ATP. Okay. So this is going back to the 3 primary things we get out of chapter 15. Okay. ATP, NAD, and FAD. Recognizing what those structures are and where the chemistry occurs. And acetyl-CoA, sorry. Okay. And then how those things are used is our energetics of chapter 15. Okay. Questions on that? One thing that I noticed when going through and looking at this practice exam is that it's a lot of just facts. There isn't a whole lot of kind of application to anything. And you can tell, at least by that question 3, I'm trying desperately to get away from facts and get at, can you interpret what's happening in this new system? Okay. But it's a lot of just raw facts. Okay. It's kind of unfortunate, but that's the way this section falls down. Starches, all of this kind of stuff. We did that all on exam 2, right? We went after exam 2. We went straight into 15, didn't we? With our energetics. So we can ignore any questions on starches. Technically, you're responsible for it. If I go through and write the exam, because it still hasn't been written yet, I'm hoping to have it done by tonight. If I go through and write it and I'm at like 20 questions, I really don't like that. Okay? Because that means if you make a mistake, you're screwed. So I might go back and add questions from older material as a review. I'm going to try to avoid that because I don't like doing that in a class where it's not necessary to do so. Okay? So just be aware of that. If I do pull back from older stuff, I'll let you know where I'm pulling back from.
Just as long as you put a little ANS at the bottom.
I might be able to do that. We'll see. I mean, if you do look at the front, I don't know if anybody's, well, some people should have noticed this. If you have to guess, then answer choice E. Anybody seen that?
You appreciate that.
Yeah. It's true. I do. I actually go through and count, painstakingly count each of them and make sure it adds up appropriately. More starch, more starch, more
Wait,
starch.
What's the answer for 4?
For 4? Oh. For 4? We just did all the labeling.
No, for multiple choice. It's a starch question, not Irish.
Oh, you just wanted to know the answer?
Yeah. It was like A or E.
Increasing hydrogen bonding interactions. Think about your chitin and your bacterial cell walls. What are they for? Structure. Right, which means, what do we want between our carbohydrates? A lot of strong intermolecular forces. Hydrogen bonding is your biggest difference there. So if you know what those structures are, you could then reference it and say what's going on with it. But it's primarily just increasing intermolecular forces. So this is a question that kind of goes from memorization and kind of goes from applying intermolecular forces. Okay. Okay, so now officially into useful stuff or relevant stuff for this exam. Which of the following statements is true for catabolic processes? What is happening for catabolism?
Generally, we're releasing some energy.
Break stuff. Okay. And by breaking stuff, what are we doing? Getting access to the energy within that stuff. Okay. So if we look through our answer choices, okay, endothermic means endothermic is absorb. That's not right. Involves synthesis of larger molecules. You're building stuff. That's not right. Organic material is oxidized. That's a nasty question. So I would skip that and move on. Involves the acceptance of electrons. Okay. Also a nasty question, so I'd be a little bit more careful on what that's saying. Acceptance of electrons is gaining electrons. That's reduced. Okay. So answer C and D are counter to each other. And E requires ATP conversion to ADP. No, so that one's wrong as well. So we're now up in that oxidized versus reduced. Oh, God. In our catabolic process, we're tearing things down.
Usually oxidizing things, right? So we're taking an electron, giving it to something else like NAD, breaking something in the process.
NAD to NADH is Yeah, you are producing NADH, and you did that by tearing glucose apart. So,
But you also built NADH up, though.
what's that?
You also built the other molecule up.
And this comes, so regardless, if you do an oxidation reaction, you are also doing a reduction. So it's going to come down to which part are you looking at. We are tearing apart glucose to make energy. Okay? So we're tearing apart glucose to make NADH. So it's catabolic, we're destroying glucose to make our NADH. What did we do to the glucose? Okay? We gave up electrons to our NAD+. Okay, which means the glucose was oxidized. I'll make sure I'm not screwing anything up. Where are we? Yes, we got that right. Does that make sense? Okay, so, and that is painful, because when you look at oxidation and reduction, it is very hard to kind of separate those. That's why it's saying very specifically our organic material, and even that is misleading because it's in biochem, everything is organic. Okay? So we have to be careful with that referencing. Okay? The other approach to this would be the memorization approach. There was a slide, which I'm pretty sure I can find. Oh crap, but I went too far back and it freaked out the system. If we pull up the slides, we've got it already specified. Okay. So it's kind of a straight from the slide thing. And you'll notice that when we just answered it, what did we do? We reasoned our way through it. That's the process I like. I didn't memorize this. Okay. So different approaches to different ways to get through it. Reasoning through is going to take a hell of a lot more time than just having it memorized. Does that make sense? In theory, we get the exact same answer. In theory. 6. Following a reaction, what species is reduced? Can't say I'm a fan of this question, but I will say the reason it's showing up is because I'm out of questions to ask. I got too few questions in the exam, and I didn't like it, so I started throwing in other kind of nonsense questions. Okay, it's not strictly nonsense because we did talk about this. We go back to our slides. The exact reaction was in the slides. Okay, so if you're reviewing the slides and how you process through and do it, you should be able to figure it out. Okay, so what is the thought process for approaching it?
If you look at carbon monoxide, it gains an oxygen, so you can say that one was oxidized. I think that was kind of an easier thing to see. So if that's being oxidized, then one of our other species, probably I2O5, is being reduced.
So if carbon monoxide was oxidized, then what was reduced? You want to volunteer it again?
So are you
The iodine species
the I2O5?
was reduced. Where's our iodine species? It's the I2O5. That's the species that was reduced. Okay. What is it reduced to? It was reduced to I2.
So by saying the species, that's like saying like the oxidizing agent, because you're,
The molecule.
No, because if you're saying reduced, it's the species that was reduced. I2O5 was reduced. What did it end at? The question actually isn't even asking that. It's just saying it was reduced. I2O5 dropped in oxidation state, or a piece of it dropped. So I2O5 is our answer there. Okay. Process of converting an alcohol to a carbonyl. NAD plus acts as A or N by blank 2 electrons. Okay. So I would again write out an equation. NAD plus then goes to what? NADH. To do that, what happened? it had to gain 2 electrons, which means it was reduced. Check your answers.
Yeah, but it's asking, it's an agent, and it's opposite, so it's oxidizing agent.
Yes,
and
that's what I was getting at. So we're identifying what changed. It was reduced. This is where I would hope and pray that we don't see an ing or an ant in the answer, and unfortunately we see lots of ings. Okay? Which means we're looking at the opposite of what actually happened. So if it was reduced, it was the oxidizing agent, which gets weird because if it's the oxidizing agent, it was reduced, which means it gained or accepted 2 electrons. Make sense? So we have to be very careful in that approach, and you'll notice that I drew out a whole bunch of stuff. Leanna, I don't know if you wrote out a bunch of stuff. It sounded like you just kind of knew all that, which, good job. Okay. I would write it out, process it, and get the definitions down because it is very easy to get those confused. Okay. So what did we say our answer was there?
A.
Oxidizing agent and accepting. So we got our A. I need to clear some space up here. Which of the following describe why the conversion of ATP to ADP releases energy? Which is really a straight from the homework question. Okay. A free phosphate on its own has resonance. Okay. By being resonance stabilized, it's more stable, so that means your products are lower in energy than your reactants. Well, that's looking at a release of energy, so that makes sense. Ion repulsion in the triphosphate. So what we're saying is when we look at ATP, what's happening? So there's our A, here's our first phosphate, second phosphate, third phosphate. When we look at those phosphates, there's a negative charge on those. And yes, there's O's and other stuff in there. Okay. But those negative charges are pushing away from each other. In ATP, they are forced next to each other, which then means they want to get apart. So we're putting them in a very high energy state. Okay? So that, again, ion repulsion in the triphosphate puts the ATP at a higher energy level, which then means more energy released. That makes sense. Entropy increases. Also an equally obnoxious question. We get a lot of obnoxious questions in this unit. Whoops.
You have more molecules here, so there's more.
We have more molecules on the right-hand side of the equation, which means more disorder. Entropy definitely increases in that conversion process. Easy way to switch the question?
Go from ADP to ATP.
Go ADP to ATP. Okay. Use the same kind of thought process in approaching each of those pieces. Make sense? Dang it, we are not going anywhere near as fast as I thought we could. Which of the following are used as electron carriers? 2 and 3. FAD and NAD. Well, technically FADH2 and NADH. What if I'd written NAD plus FADH2 and NADH?
Wait, say it again.
What if I'd included, instead of ATP, sure, actually let's do that. ATP, FADH2, NAD plus. This would be kind of a mean way to do it, but we could approach that.
Just 3.
Is NAD plus carrying any electrons?
No, but it can. It's acceptor. Oh,
Okay.
it's a carrier.
It's not an electron carrier at this point because it doesn't have any electrons. Okay. Be careful if you see a question like that. Just be like, are you really doing this to me?
So then it's like none of them.
In that case, it would be none of those. None of those are carrying electrons. Okay. I would probably rephrase the question to which of the following are carrying electrons. Okay. Instead of being electron carriers. Because NAD plus has the potential to carry electrons, which is where it becomes questionable. Changing the question to which of them are carrying electrons, NAD plus is not carrying electrons. Okay, it can, but it doesn't have them. Does that make sense? Which of the following reactions is spontaneous? What does spontaneous mean?
Negative G.
Delta G must be negative. Yeah. That's what I want to do. Delta G is less than 0. Okay, so any of them that are negative, B, there we go. Spontaneous. Woohoo. 11, which of the following processes is an example of oxidation? Okay. For those of you that are good with your definitions, screw you. I would take oxidation and I would write underneath it loss of electrons and try and apply that loss of electrons into each of these equations. Okay. Looking at those, I would probably also tag an extra little piece. Gain of oxygen or a loss of hydrogen. Okay. So I would start with loss of electrons. I would look at those and I would be, oh crap, I don't know the oxidation numbers. I need a quick rule of thumb. Increase oxygen, decrease hydrogen. ADP to ATP, you should immediately eliminate. There are no electron transfer reactions in ATP. Okay. Keep that out of the back of your mind. NO2 to NH3, what did we do? We gained hydrogen. That's not oxidation. NAD plus to NADH. We again gained hydrogen. ADP, being obnoxious again. Okay, what happened in E?
Lost hydrogens.
If I didn't gain oxygen, also being obnoxious. Okay, both increasing oxygen and decreasing hydrogen are valid on their own. E is your answer. Make sense? Determine the free energy change for the phosphorylation of glucose in the first step of glycolysis.
So you did it backwards, because we go from ATP to ADP, so you did that because you didn't want to put the negative, because you wanted to be ornery, so that we'd make a dumb mistake, but that's what it's supposed to be, right? It's negative 30.5, if you go the other way.
Yeah. So I'm actually surprised, because I do tend to be ornery, as you're pointing it out, that I actually didn't include 13.8.
Just to do, yeah.
Because if we look at what the question's asking, the free energy change for the phosphorylation of glucose, what's happening in that very first reaction? Phosphorylizing glucose. 13.8 is a perfectly valid answer for part of the question. What's the other part of the question?
It's the whole step. You said
The first step of glycolysis. And in the first step of glycolysis, are we putting a phosphate, a free phosphate, onto glucose? No, we're using ATP. Okay? So it's getting you to recognize that to get that first step to happen, I need more energy. I need a source of the phosphate. I need to couple that with the ATP consumption, which needs to happen in the reverse. ATP goes to ADP plus our free phosphate. Okay, we could add water if we really cared. Okay. Since we are reversing the reaction, what happens to the delta G?
It's a negative.
It goes negative. It's pulling our knowledge from 151 General Chem. We can then add our two terms together, get rid of that, and we end up with what, negative 17-ish? Did I do that math right? Hey, look at that. My math wasn't too far off. We end up with C. You're more than welcome to use a calculator. I didn't need a calculator to go through and do this. I'm not going to be putting rounding issues in here. You're looking at the big differences in those numbers. Make sense? In glycolysis, ATP is synthesized by...
Substrate level phosphorylation.
Okay. We need some kind of phosphorylation, and look at that. I had to make up some kind of terms to do some phosphorylations in there. What is photophosphorylation? Light is somehow being involved. I'm pretty sure I just made that up.
It It probably is
Okay.
in chlorophyll. I
In
bet you it
chlorophyll, they may reference it, the formation of ATP in chlorophyll
does. I think it is.
might be called photophosphorylation. Okay. But it's using light. Okay. Well, glycolysis doesn't involve light. We're doing the substrate level phosphorylation. Okay. Oxidative phosphorylation is a bit weird because they're referencing the electron transport chain. Okay. So glycolysis hasn't done that. What would be the limiting factor for ATP production by glycolysis in a strenuously exercising muscle that lacks lactate dehydrogenase?
NAD+.
It's our NAD+. Yes. Yeah, I was going to start answering NADH, but no, it's the NAD+. So, and that...
Because it's the other pathway, because you said
Yep.
glycolysis.
In a strenuously exercising muscle, are we getting oxygen?
No.
No. So we need to produce energy as fast as possible. So that means running glycolysis. To run glycolysis, we need to dump electrons out. Okay? That is our NADH. Okay? Our NAD+ needs to act as an electron acceptor so that we can process through that. Okay? If we don't have NAD+, glycolysis shuts down. And it shuts down early on. It shuts down before we actually produce ATP. Okay? So if we don't have lactate dehydrogenase, well, now what happens?
We just have NADH.
I have
You can't
NADH.
do anything with it.
And I can't do anything with NADH if I don't have oxygen. So that's the strenuously exercising part of this. Okay? We're now shifting into the anaerobic conditions. Well, I need something to convert that NADH across. That's the goal of the lactate dehydrogenase. Okay? Is to take that NADH that we made and say, oh crap, I can't get efficient energy usage out of it. I'm going to reverse that reaction to make lactic acid. Okay? Now I have NAD+ that I can shuffle back into glycolysis and initiate and produce a couple more ATP. Okay? So NAD+ is kind of a key intermediate in our glycolysis to initiating and keeping that reaction running. Enzymes responsible for the following reaction. Told you this kind of nonsense question would show up. Okay? A variety of ways to do it. Based on how quickly you guys answered, it's because you guys remembered it. Yeah. Okay? You can go through and memorize the enzymes, that's fine. The other approach, look at what changed. What's the big change? We have a phosphate group added. What enzyme puts phosphates on? Kinases. Okay? Being a bit mean because we've got that phosphorylase name in there, getting you to think along those lines. It's just tying the names of the enzymes back to what they do. Okay. Formation of glyceraldehyde 3-phosphate from glucose. Okay. Formation. Oh, this will be fun. Yes, 2 ATP are consumed. Okay. And that is going to be kind of one of our big things because it's now looking at a section of glycolysis, not the whole thing. If we look at the whole thing, what's the result?
4
I'll kind of accept 4 produced. What I would have to add if we said the whole thing, how much ATP is produced, I would probably add the word net.
So
It's
net
not.
2?
It's net 2 because 2 are consumed. Okay.
Oh, really 4.
So 4 are produced. Again, it's going to come down to the level of the question. Because you're doing the MCAT, Mike, this is a question that you should be careful on. Is be really careful on what they're saying. If they say a net produced, it's 2. Okay. If they're saying what is the overall or the total produced, it's 4. You have to be careful with your question. Pyruvate decarboxylase, large enzyme, yada, yada, yada. Thiamine is important. What does thiamine do? It is an electron acceptor and an electron donor. Okay, so A and B. Sorry, got a little confused with the words. So A and B are our big pieces there. Bit misleading because we have proton donor in there as well. It can, to act appropriately, it has to donate a proton, but it's probably donating that proton to the enzyme, not to the substrate. Okay. The other way it could be asked is thiamine is similar to. Will I accept that? I don't think I'll accept that. No, that's a lie. I'd accept that. It is going to do a similar function. Okay. But for different reasons. So I would kind of accept that. Though vaguely. You should be tying cysteine to coenzyme A.
Okay.
Cysteine and coenzyme A should be tied together. Cysteine to thiamine is questionable. Okay. What is the similar thing between cysteine and CoA? The sulfur group. That's the thiol. Have you heard people say thiol?
I don't know, I just tried in my head, that's how it said.
I've always said it thiol, and I have no idea. I mean, there's some people that call them carbonyls, and I always thought that was really weird. I have a really hard time keeping a straight face when someone calls them carbonyls, but it's a standard way to say it. So, better answer for thiamine. It is TPP. Carbene. Thiamine is your biochemical carbene. Which of the following are true? You can always watch out for me stupidly leaving you the answers. So the answer to this one is that all of these reasons are why the citric acid cycle is considered your central metabolic pathway. Okay. Metabolism of sugar and amino acids. Okay, this is looking at sugars, glycolysis. How do we use the products of glycolysis? Krebs cycle. So immediately involved. Okay, amino acids and lipids.
That was the anabolic.
That's potentially the anabolic, and we can reverse. All of those processes can reverse. So that's why we're saying metabolism. Okay, if we go through and look at those figures again, we can go out and in from each of those directions. It links anaerobic to aerobic. Well, the anaerobic was all glycolysis. Aerobic is the electron transport chain. So the products that come out of glycolysis go into the Krebs cycle to get us into into our electron transport chain. D is pretty much the same thing as A and B, except more general. Make sense? Following mechanism, there's got to be a better way for me to put this question together, because I think this looks kind of confusing, personally. So what I would do is get a piece of paper, cover all that up. Okay. So we have a plausible mechanism for the conversion of the acetone to our glyceraldehyde. What we're now asking is, what is the enzyme doing? This should look kind of familiar. You've seen a question like this in, I think, the previous exam. Okay. So we're looking at the enzyme. What does the enzyme need to supply?
Electrons.
We need electrons. We need something that has some base characteristics to pick up that hydrogen. So it's now then looking at the rest of those structures to decide. Oh, come on. Why are you not scrolling? Okay. And you'll notice that we have a negative charge on that enzyme. So NADH does pick up hydrogen. Does it pick up a positive hydrogen? No. In this case, we're not using NADH. We're picking up an acid. So NADH is out. Glutamic acid, hey, we got a negative oxygen. That could work. But there's a negative oxygen in histidine and in arginine and in leucine. So C and D are basic amino acids, but look back at your drawing. The enzyme has what charge?
Negative.
A negative. So why are you saying C and D over B and E, Jimmy?
Oh, I guess I was looking at them as accepting a positive hydrogen
They could accept, but they aren't negatively charged, so that doesn't match the drawing. Okay, so C and D become questionable with their R
You're
groups.
saying that one of these things down here is that enzyme we're talking about?
Yep,
Oh,
Would you say B?
it's going to be B. Why are we ignoring the negative charge on most of those structures, say on the arginine, the histidine, and the leucine? Why do we ignore that negative oxygen?
Balanced out with the positive.
Nope. Protein backbone.
So it's D.
It's occupied in the protein backbone. That negative oxygen isn't there that I've got circled. Those are in the protein backbone. We only look at the R group. The R group is where our functionality is located. So this is a way of me pulling back at old material to get you to think about that old stuff back in the enzymes.
so we're saying